Inside your body exists a genetic system scientists still do not fully understand. For decades, most of your DNA was called useless. But what if that was completely wrong? What if hidden inside your genome are ancient human species, biological control systems, and mechanisms that are still changing you right now? For decades, scientists looked at the human genome and believed they understood its structure. But when they started analyzing it in detail, they ran into a problem. Only a very small portion of DNA directly produces proteins. So the remaining majority was labeled as junk DNA, not because it was proven useless, but because its function was unknown. For years, it was treated like biological noise, leftover material from evolution. But that assumption started to collapse in the early 2000s. A major turning point came with large-scale international research efforts like the ENCODE Project, which began publishing results around 2012. ENCODE stood for Encyclopedia of DNA Elements. Its goal was simple in theory, but extremely complex in practice, to map what every part of human DNA actually does. And what they discovered changed the way scientists think about the genome. They found that a large portion of non-coding DNA is actively involved in biological regulation, not in building proteins directly, but in controlling how, when, and where genes are activated. These regions act like molecular switches. Some sequences function as enhancers, increasing gene activity. Others act as silencers, reducing or completely shutting down gene expression. There are also promoter regions, which determine when a gene starts being read. And what makes this even more complex is that these regulatory elements are not always close to the genes they control. In some cases, they can be located thousands or even millions of DNA base pairs away. Yet through the folding structure of DNA inside the nucleus, they physically interact. This means that DNA is not a simple linear sequence. Inside the cell, it behaves more like a folded 3D structure where distant regions can touch and communicate. So instead of a straight line of instructions, you are looking at a three-dimensional control system operating inside every cell, and this system is constantly active. It decides whether a cell becomes a neuron in your brain, a skin cell on your surface, or a muscle fiber in your body. All of this depends not only on the genes themselves, but on how those genes are regulated. And this is where biology becomes even more complicated, because the same gene can behave differently depending on which regulatory elements are active. Meaning, two cells in your body can contain the exact same DNA, but produce completely different outcomes. A neuron and a skin cell share the same genetic code, but express it in entirely different ways. This is called gene expression control, and it is one of the core principles of modern biology. But despite decades of research, scientists openly admit something important. Even after ENCODE and other genome mapping projects, we have only identified the function of a fraction of these non-coding regions. A significant portion of the genome is still not fully understood. We can see where it is. We can measure it. But we cannot yet fully explain what it is doing at every moment inside a living human body. And that leads to a simple but powerful realization. The junk DNA was never junk. It was simply a system we were not advanced enough to understand yet. For most of human history, we believed we were the only surviving human species. But modern genetics revealed something completely unexpected. Around 50,000 to 60,000 years ago, early Homo sapiens began migrating out of Africa into Eurasia. And there, they encountered other human species that were already living in those regions. Most importantly, Neanderthals in Europe and Western Asia, and later discovered Denisovans in parts of Asia. For a long time, these were considered completely separate branches of the human family tree, extinct cousins. But when scientists began sequencing ancient DNA from fossils, they discovered something shocking. These species did not just coexist with us, they interbred with us, which means their genetic material did not disappear. It was passed on, and it is still present in modern humans today. On average, people of non-African descent carry about 1 to 2% Neanderthal DNA. In some populations in Asia and Oceania, there are also traces of Denisovan DNA, in certain cases even higher in specific genetic regions. This is not symbolic, it is measurable in your genome. And these fragments are not inactive remnants, they still influence biology today. For example, studies have linked Neanderthal-derived genes to immune system strength and how the body responds to viruses, skin and hair traits adapted to different climates, and even certain neurological and metabolic differences. In other words, some parts of your biological response system are inherited from extinct human species. This completely changes how we understand human identity, because it means you are not the result of a single uninterrupted lineage. You are a genetic mixture of multiple human species that once shared the Earth. And this story becomes even more fascinating when we look deeper into Denisovans. Unlike Neanderthals, we do not even have a complete skeleton of Denisovans. They were first identified not through bones, but through a tiny fragment of finger bone and a few teeth found in a cave in Siberia. Yet from this minimal evidence, scientists were able to reconstruct an entire genetic profile. And what they found showed that Denisovan DNA helped modern humans adapt to extreme environments. One of the most famous examples is found in Tibetan populations. A specific gene variant associated with high-altitude oxygen processing has been traced back to Denisovan ancestry. This allows the body to function efficiently in low-oxygen environments like the Himalayas. So in a very real sense, part of your survival system may come from a human species that lived tens of thousands of years ago in environments completely different from your own. And this leads to a deeper realization. The human genome is not just a biological instruction set, it is a historical archive of survival. Every fragment of ancient DNA inside you represents an adaptation that once helped someone survive a different world. Cold climates, new viruses, low oxygen, changing environments, all of it is still written into your biology. But here is what scientists are still trying to fully understand. We know these fragments exist. We know some of their effects, but we still do not fully understand the complete interaction between ancient genes and modern human biology. Because these genes do not work in isolation. They interact with thousands of other genetic and regulatory systems inside your body. So the question is no longer just what did we inherit, but rather, how is that inheritance still shaping who we are today?